Hello everyone, welcome back to my channel. So today we are going to study about epigenetic editing. So before proceeding towards the video, I just want to request to you all to please like, comment, share and subscribe to my channel. So let's get started. Let's understand epigenetic editing, or which refers to as redefining gene expression without changing the DNA sequence. So editing, we all know what is editing. Now editing of a gene is epigenetic editing, and the study of it is epigenetics in a very layman term. So we are going to study today what and how it is benefiting us. So let's begin with the basics. What is epigenetics? So epigenetics is the study of the changes in gene expression that occur without altering the DNA sequence itself. डीएनए सीक्वेंस को चेंज किए बिना, ऑल्टर किए बिना, जीन एक्सप्रेशन में जो हम चेंजेस होते हैं जो जीन एक्सप्रेशन के, उसकी जो हम स्टडी करते हैं, जीन कोई भी पर्टिकुलर जीन इस तरीके से एक्सप्रेस हो रहा है या उसके साथ फंक्शन कर रहा है, व्हाटएवर वी आर स्टडीइंग विदाउट ऑल्टरिंग द डीएनए सीक्वेंस इटसेल्फ, दैट study is known as epigenetics. Now there is a huge difference between genetics and epigenetics. So see, genetics is more of like a hardware of a computer, and epigenetics is the software that controls how the hardware runs. So there is this huge difference. Genetics is basically the outer part of a body, and epigenetics is the inner part. Okay, and it is controlling the outer part. So that is how they both are interlinked. Now main mechanisms in studying epigenetics: DNA methylation, histone modification, chromatin remodeling. ये कुछ main mechanisms हैं. These changes act like switches that turn genes on or off depending on the needs of the cells. अभी ये जो mechanisms होते हैं, they either switch on the gene expression or switch off the gene expression depending on requirement क्या है cell की, तो उस हिसाब से वो gene expression on and off होता रहता है, and these are some of the mechanisms that helps the cell doing it. Okay, moving forward to what is epigenetic editing. Now epigenetic editing क्या होती है? So epigenetic editing is a powerful new approach where we make targeted and reversible modifications to the epigenome. So epigenetic editing जो होती है एक new approach है. It's a new approach where epigenetic editing is a powerful approach जहाँ पर हम epigenome में targeted and reversible modifications कर सकते हैं. reversible यानी कि वो it is reversible, वो वापस से back जा सकते हैं, and targeted यानी कि हम वो particular उसको target किया जा सकता है. So targeted and reversible modifications to the epigenome. Unlike genome editing, which changes the DNA sequence permanently, epigenetic editing only alters the chemical marks on DNA or histones. DNA genome editing में क्या होता है, DNA sequence alter हो जाता है. epigenetic editing में वैसा नहीं होता. chemical marks सिर्फ alter होते हैं. DNA या histones पे जो भी chemical marks होते हैं जो gene expression के, वो सिर्फ उसको alter किया जाता है, and that is also reversible. It is targeted, हम टारगेट करते हैं स्पेसिफिक सीक्वेंस को और वो रिवर्सिबल होता है, एंड वो पर्टिकुलर डीएनए मार्क के केमिकल मार्क को ही टारगेट करते हैं सिर्फ. ठीक है, ये पूरा डीएनए सीक्वेंस को ऑल्टर नहीं करता है. सो इट इज डिफरेंट फ्रॉम जीनोम एडिटिंग इन दैट मैनर. जीनोम एडिटिंग जो कि क्रिस्पर्स, टेलेंस, ZFNs इन सब से की जाती है, दैट इज जीनोम एडिटिंग. ओके, सो epigenetic editing in the longer run allows the precise control of gene expression without introducing mutations. Precise control allow करता है gene expression का. gene expression को precisely control किया जा सकता है with the help of epigenetic editing without introducing mutations. बिना mutations के उसको properly control किया जा सकता है, which makes it a safer and flexible tool to use. So the process follows a flow, starting with targeting a specific site, then binding to that specific site, followed by the modification, and finally the regulation of gene activity. ओके सो इसमें एक प्रिसाइज कंट्रोल होता है जीन एक्सप्रेशन पे. म्यूटेशंस नहीं हम कॉज कर रहे हैं, किसी भी डीएनए लाइक सीक्वेंस को ऑल्टर नहीं कर रहे हैं. सो एक प्रिसाइज कंट्रोल रहता है एंड दैट इज वी फर्स्ट टारगेट, देन वी बाइंड, देन मॉडिफाई एंड उस तरीके से भी जीन एक्सप्रेशन रेगुलेट होता है. ओके सो इट इज एज सिंपल एज स्टेटेड. Moving forward to molecular tools for epigenetic editing. Now what are the molecular tools? So several molecular tools make epigenetic editing possible. CRISPR-Cas9 systems are the most widely used components which makes epigenetic editing successful. So what happens in this is a deactivated Cas9 protein, जो आपको Cas9 दिख रहा है ना, so that deactivated Cas9 protein is fused with enzymes that add or remove epigenetic marks. Cas9 जो होता है, deactivated Cas9, वो enzymes के साथ fuse करता है, तो या तो वो add करता है epigenetic marks को या फिर वो उसको remove करता है. So that is how CRISPR-Cas9 systems help in epigenetic editing. Okay. Zinc finger proteins जो होते हैं, तो zinc finger protein या TEL effectors, they also can be engineered to target specific sequences, epigenetic marks को remove करने के लिए या add करने के लिए. These proteins are often fused with enzymes. ये proteins को फिर further fuse किया जाता है enzymes के साथ. So the enzymes can be DNA methyltransferases, written as DNMTs, DNA methyltransferases, TET enzymes, histone acetyltransferases, histone deacetylases to perform the editing. इनके साथ उसको bind करते हैं, इनके साथ fuse करते हैं enzymes को. That's how they function. Each tool has its own advantages and limitations, which researchers choose based on the application, based on what they are performing. Okay. Moving forward to mechanism of action. किस तरीके से मेन मोड क्या है फंक्शनिंग का एंड किस तरीके से ये होता है प्रोसीजर. सो सी डीएनए मिथाइलेशन एंड डीमिथाइलेशन विच जनरली साइलेंस और एक्टिवेट द जीन्स. ये फर्स्ट स्टेप होता है कि डीएनए मिथाइलेशन किया फिर डीमिथाइलेशन किया, तो वो बार बार बिकॉज सिक्वेंशियल मिथाइलेशन एंड डीमिथाइलेशन की वजह से क्या होता है, जीन्स जो है अगर आप मिथाइलेशन कर डिपेंड्स, अब आप मिथाइलेशन कर रहे हो या आप डीमिथाइलेशन कर रहे हो, तो उसकी वजह से या तो जीन साइलेंस हो जाएंगे या फिर जीन एक्टिवेट हो जाएंगे, डिपेंडिंग ऑन व्हाट यू आर डूइंग एंड व्हाट यू वांट टू डू. सो जीन्स उसकी वजह से साइलेंस या एक्टिवेट हो जाते हैं. देन हिस्टोन एसिटाइलेशन एंड डीएसिटाइलेशन which loosen or tighten the chromatin to regulate gene accessibility. Again it depends on acetylate कर रहे हो या deacetylate कर रहे हो, तो उस basis पे वो lose हो जाएगा या वो tight हो जाएगा. based on अगर आप acetylation कर रहे हो तो वो lose हो जाएगा and based on deacetylation कर रहे हो तो वो tight हो जाएगा. So that's how gene accessibility बढ़ती है. अगर आप उसको lose कर दोगे तो gene accessible हो जाता है, tight कर दोगे तो gene उतना accessible मतलब आप रेगुलेट कर सकते हो. सी ऑल इट इज ऑल if you want to remove the epigenetic mark or if you want to add the epigenetic mark. So these are some of the mechanism how to do it, which helps us. Chromatin remodeling, chromatin state modulation या chromatin remodeling. So it changes the overall structure of DNA packaging. पूरा जो structure है उसको change कर देता है वो. In short, this process can be summarized as again targeting, then binding, then modification, and further gene regulation at the end. Okay, so that's how this particular mechanism works. Okay, moving forward to application of epigenetic editing. अब क्या applications हैं? How do we perform this process and how is it beneficial for the whole thing? So epigenetic editing has exciting applications across fields. Epigenetic editing जो है बहुत सारे उसके benefits हैं. So epigenetic editing has exciting applications across fields. So in cancer therapy it can reactivate tumor suppressor genes that were silenced. जो भी tumor suppressor genes होते हैं जो tumor को suppress करते हैं वो genes को regulate कर सकता है cancer therapy. So it benefits in the cancer therapy. इन न्यूरोलॉजिकल डिजीजेस में जैसे कि अल्जाइमर्स है, हंटिंगटन्स है, इट कैन रीस्टोर नॉर्मल जीन फंक्शन को रीस्टोर किया जा सकता है, दैट इज पॉसिबल. सो इन न्यूरोलॉजिकल डिजीजेस सच एज अल्जाइमर्स और हंटिंगटन्स नॉर्मल जीन फंक्शन को रिस्टोर करने में हेल्प करता है. It is also being explored in autoimmune and metabolic disorders. Currently, it is in research. In regenerative medicines, epigenetic editing helps, reprogramming of the stem cells for tissue repair. Tissue repair के लिए वो reprogramming करने में help करता है. And agriculture में it can improve crop yield and stress tolerance by turning gene expression. अगर gene expression को tune कर दिया जाए, turn कर दिया जाए, जो भी gene expression है, if it is helping तो उसको particular add कर सकते हैं, and if it is not helping उसे remove कर सकते हैं, whatever is the case. but वो करके agriculture में भी वो yield बढ़ाई जा सकती है, तो it will give more yield and will improve the crop yield. So उस तरीके से it can also benefit. Okay, now what are the advantages of epigenetic editing over genome editing? So first is it is reversible and tunable. It is modifications किए जा सकते हैं and reversible है. That is first advantage. We can adjust gene expression like a dimmer switch instead of making permanent changes. Permanent change नहीं करके हम small changes से जो हमारा aim है वो हम हमें मिल सकता है, that is possible. So that is first advantage. Second, it does not alter the DNA sequence, so the risk of mutations are minimized because again mutations alter नहीं हो रहा है तो mutations नहीं होंगे. Third, it allows dynamic control. Genes can be turned on in one situation and off in another. So it is providing us wide variety of options to do something, because it is not providing just one option, it is providing two options, whether you want to silence the gene, whether you want to activate the gene. So वो बहुत ही wide applications खोल देता है इसके लिए. Okay, so these unique advantages make epigenetic editing particularly attractive for therapeutic applications. Okay, next is what are the challenges and ethical concerns with this. So of course there are challenges, because it is an emerging trend, so of course there are going to be challenges. Sometimes these editing tools can affect unintended targets जिसे हम target नहीं कर रहे हैं, which can lead to off target modifications. Long-term stability of epigenetic changes is also a concern, because will the changes persist or fade over time, वो भी एक concern है. Another challenge is the delivery system. Delivery system is also one challenge. How do we get these editing complexes safely into human cells? कैसे हम transfer करेंगे human cells के अंदर? So that is also one challenge. And finally, ethical concerns तो हैं ही हैं, especially when we talk about potential germline editing changes that could be passed on to future generations. So that is one important concern. Now, what are the future trends in epigenetic editing? So looking ahead, epigenetic editing will play a key role in precision medicine, allowing customized treatments based on a patient's epigenome. It will also be combined with other therapies, for example, pairing CRISPR with epigenetic modulators. Artificial intelligence is expected to guide predictions of epigenetic states and epigenetic outcomes, editing outcomes. And with ongoing clinical trials, we may soon see approved therapies that use epigenetic editing as a mainstream medical tool for, several applications. Okay, a quick recap to summarize: epigenetics control gene activity without changing DNA sequence. It also allows us dynamic control by providing, the switching on and switching off of the gene expression. Epigenetic editing also allows precise, reversible, and tunable regulations of genes. It has vast applications in medicine, agriculture, biotechnology, and with challenges remains, the future looks very promising, especially in the field of precision medicine and personalized treatment. So उस तरीके से ये है. So thank you so much for watching. I hope you enjoyed the video. If you found this video useful, please don't forget to like, comment, share and subscribe to my channel. And if you want any particular topic of your choice to be explained by me, please do let me know below in the comment section. I'll definitely make a separate video onto it. Okay, so till then, bye bye, take care. Thank you so much for tuning in. Bye bye, take care. We'll meet in the next video.